Hello, my name is Maxwell Esiedu. I'm a PhD student in plant biology. On this channel, I would like to introduce you to various bioinformatic tools I routinely use during my research. So I've come across a number of students who had challenges with various tools that I mostly use during my research. So I thought it would be useful that I create this channel to talk about my experiences and my insights in these tools that I use. So in this first video, I would like to talk about GWAS, which is an abbreviation for Genome-Wide Association Studies. So GWAS refers to the association of genetic variations within the genome, preferably single nucleotide polymorphisms, with the traits that you may be working on. All right. So in essence, GWAS allows us to identify the genes that may be responsible for controlling the trait. That's a simple explanation of what GWAS does. All right. So to perform a GWAS, you would need a population. All right. So I am working in the plant sciences, specifically root biology. So let's say I want to understand what are the genes controlling the root diameter, the root angle, root length, etc. What I'll do is to get a population of lines or genotypes or plants, all right? And then I'll phenotype the plant for the specific trait I'm working with. In contrast, if you are working in medical sciences or other areas of biology, the same principle applies. All you need is to phenotype the population to get your phenotypic data. All right, and then I'll move on to genotype my plants to get the single nucleotide polymorphisms. So once you have these two key input files, you are ready to perform your GWAS. All right. So for instance, like I talked about, I want to understand. I have a population of say 200 plants or genotypes. I want to understand the genes responsible for controlling root angle in this panel. All right. So for instance, I have plant A here, and plant A has a very steeper root system architecture, as you can see. So this type of plant would allow or would be suited to areas that have abundant water within the subsoil or deep soil profiles, as well as other nutrients that may have leached into deep soil profiles. So this plant would be very useful in these type of soils. Plant B, in contrast, has a very shallow root system architecture, as you can see. So this type of plant would be suited to areas with abundant water in the topsoil, as well as immobile nutrients like phosphorus within the topsoil. So what I will do if I'm performing the GWAS is to phenotype all the lines in the panel for these root system architecture. So when I say phenotype, what I mean is I would measure the angles, as you can see here. So for instance, let's say this root or this plant A has an angle of 40 degrees, plant B has an angle of 100 degrees. So I'm going to measure all the angles for all the lines in the panel just like this, and that becomes my phenotypic data. And then I would move on to do the genotyping of the population. All right. So for instance, plant A here, so plant A has a sequence of TA, CG, G, CT, GA. Plant B has a sequence of TA, CT, CT, GA. Now the main difference in this sequence between plant A and plant B is the G here. This G is substituted for T here, in the G in A here, it's substituted for T in plant B. So this is what you are going to do when you are genotyping your population. It is going to capture all these SNPs or single nucleotide polymorphisms, and these SNPs are very much abundant within the genome. So your genotyping is going to capture all these single nucleotide polymorphism for each of the lines within the panel, and that becomes your genotypic data set or your SNPs or your SNP data set. All right. And so once we have our phenotypic and our genotypic data sets, we are ready to perform our GWAS. To perform your GWAS, there are various tools that are available. You have GAPIT and then you have Tassel. Preferably, I always use GAPIT because it has a number of modules that are very useful for performing GWAS and identifying your significant SNPs. So at the end of performing your GWAS, you will come up with this plot here, which is popularly called the Manhattan plot. All right. So what you mainly need to focus on in this Manhattan plot are the peaks that have that are more than 0.05 p-value. So all the peaks that are above the baselines you see here are what you want to focus on. All right. So for instance, you would want to focus on this SNP here on chromosome 13, this on chromosome 8, and that on chromosome 6. And what you're gonna do is to collect all these significant SNPs, annotate these SNPs, check what genes are these SNPs falling within. Are the genes these SNPs falling within annotated for biological processes that may be involved in your controlling your trait or whatever? So these are the kind of analysis you may want to do after performing your GWAS. I do not want to bore you much at this stage. I just want you to know how to phenotype and genotype your data and how to perform your GWAS. In our next videos, I'm going to take you through step by step how to perform the GWAS and how to follow up on narrowing down on your candidate gene. All right. So in summary, like I said earlier, GWAS allows you to understand the genetic factors that are controlling your trait. All right. To perform your GWAS, the key input files you would need is your phenotypic and your genotypic data sets. And then to perform your GWAS, you would need tools like Tassel, GAPIT. There are a number of other tools that you may also want to use to perform your GWAS. Like I said, in our next episodes, I will be introducing you, or I'll be walking you through the various quality control analysis you may need to perform on your input files before performing your GWAS. Okay. And then lastly, I will take you through a live tutorial on how to perform GWAS using a sample. I have a sample data set that I would like to walk you through, and then in how to perform the GWAS using GAPIT. All right. So this is the end of this video. If you really enjoyed it, or you thought that this was a useful, very educative video for you, please, I would urge you to subscribe to this channel. The next videos I'm going to upload, you get a notification once you are subscribed, and then you watch it. Thank you very much for your time, and I will see you next time. Bye.